Hi guys, um, welcome to our GCSE biology session for this afternoon. This will be the second biology session that we've done, so really excited to see lots and lots of you joining us for the session today. If you were here with us for the last session, then you will know our biology tutor, Charlotte, but if you're new, then this session will be led by Charlotte, and Charlotte has a really interesting session planned today around digestion. So if you want a bit more information about Charlotte, then she studied conservation and biodiversity at Exeter University, and she's done over 150 lessons with my tutor. All of these sessions are recorded, and you will be able to find the links to these sessions on the My Tutor online school. And how we'll run it today is, as always, 45 minutes for Charlotte to do the teaching, and then 15 minutes at the end for questions. But do send questions through to the chat in the meantime if there's anything that you want to ask. I'm going to hand over to Charlotte now, who will lead the session for us.
Okay, hi everyone. So as Frederica said, this session is about digestion. It leads on quite nicely from the last session that I led, which was about lungs and gas exchange. So if that's a topic that you're interested in, then you can go back and find that and watch that one. But today is all about digestion and how we break down complex food structures into smaller components that we can then absorb into our blood and use in our body. So we're going to talk through how that happens. There's quite a lot to get through, so we'll see how we go. And I have got some past paper questions that we can work through if needed, and as always, we can answer your questions as well. Okay, so what is digestion and what is our food made up of? Well, as we just said, digestion is when we break down complex food structures into smaller components that we can absorb into our blood and use in our bodies. And when we take in food, we take in a mixture of complex carbohydrates, proteins, and fats, or lipids. So proteins in this first row of the diagram here, we get those from foods like meat and beans, and they are made up of amino acids in a chain. So an amino acid is a monomer, and they form a peptide chain, which is a polymer of amino acids. And amino acids are useful in our body for protein synthesis, so we need those to make up the proteins and the enzymes that we use for reactions in our bodies. Carbohydrates, such as starch, are made up of lots of glucose molecules. We find starch in pasta and bread. We can't use starch itself in our bodies, but when broken down, the glucose molecules can be used in respiration to provide us with energy. And lipids are fat molecules, and they're made up of two things rather than one thing. They're made up of fatty acids and glycerol. We get those when we eat things like oil or butter, and then during digestion, they're broken down to the separate fatty acids, glycerol, and used for things like cell membranes. So if you remember any of your cell biology lessons, we talked about the phospholipid bilayer in cells. Okay, so we're going to have a quick look at our digestive system itself. So our digestive system is an example of an organ system. It's lots of organs all working together. So we start by taking food into our mouth, we're at the top, and there we mix the food with our saliva, and this begins the digestion of carbohydrates. The food then passes down our esophagus and into our stomach, and our stomach is where we begin the digestion of protein as well, and we start to absorb some of the molecules. The stomach has stomach acid in it, which helps to break down a lot of materials as well. So it's quite an acidic environment. Our food then moves from the stomach to the small intestine, and the small intestine is broken down into two areas. So the duodenum is first, and while in the duodenum, the digestion of carbohydrate and protein is continued, and then we begin to digest those lipids as well, those fat molecules. And then food passes to the small intestine, the ileum. This completes the digestion of carbohydrates, proteins, and lipids, and a lot of those smaller particles that have come from the larger particles that have been broken down are absorbed there into the blood. Then from the blood, they're transported around the body and into cells. In our large intestine, which is the next area, we have the absorption of water and then the digestion of undigested food. So that moves down to our rectum and our anus. We've also got a few more organs that are involved kind of in an outside way in digestion. So the liver produces bile, which we'll learn more about later. That helps to neutralize the stomach acid and emulsify some of those fats, but we are going to talk more about that at a later time. We've also got our gallbladder. That's where the bile is stored and then released into the small intestine to help with the digestion of food in there. And also our pancreas, which produces some enzymes and releases them to the small intestine as well. This will all make more sense when we've talked through a bit more. So how is our small intestine adapted for digestion? So most of the digestion that we talked about happens in our small intestine. And the walls of our small intestine are covered by lots of little projections. So the walls are wavy. They have all of these little, we call them projections. Their scientific name is villi. So we have lots and lots of villi. We've got a diagram of them here. So here's a villi sticking up from the wall of the intestine. It has a capillary inside it, and then the surface of the villi itself, or villus, is covered in these microvilli. So that's even smaller projections on the projections themselves. And the reason that the small intestine has these projections is to increase the rate of absorption, or to be able to facilitate lots of absorption of those small particles to happen and get those into the blood and transported around the body as quick as we can. The rate of absorption is increased by having a large surface area, a short distance, and a concentration gradient. This is very similar to the diffusion that we talked about in the last tutorial for lumps. So the villi, all projecting from the surface of the small intestine, provide that huge surface area for molecules to be absorbed over. The fact that they are each have capillaries running next to them means that there's a short distance for that absorption to take place. And because the blood is continually flowing through that capillary and taking molecules away, or particles away, to be used around the body, we have that constant concentration gradient. So there's always room for more particles to be taken into the blood. Okay. So we've said a lot about food being broken down into those from larger particles into smaller particles that can be then used in the body. But how does that happen? So digestion happens using enzymes. They're a really key part of this process. Enzymes are biological catalysts. They help us with reactions in the body. And they are proteins as well. So they are made up of amino acids. And each enzyme has its own particular structure. And this allows it to do its own particular tasks, so to help its own particular reaction take place. And when we talk about enzymes, we talk about the lock and key mechanism. So each enzyme has a particular shape to fit with a particular substrate particle. So if we follow the diagram through here, number 1, the substrate, which in this case would be our starch or our lipids or any of the proteins that we talked about in that first slide, collides with the active site of the enzyme and becomes attached. An active enzyme or an active site on an enzyme is the particular part of that structure of the enzyme that fits with the substrate. So it's this section here. The active site has a complementary shape to the substrate. So you can see here that the enzyme and the substrate fit together like a lock and key. Complementary is a key word here. We need to remember that. It's useful in all sorts of topics. So here the active site has a complementary shape to the substrate. Number 2, the enzyme catalyzes the breakdown of the substrate. So the fit between the enzyme and substrate is like a lock and key, so they're now attached together, and the enzyme can then, now it's got a good grip on that substrate, that particle that it wants to break down, it can then pull it apart. So the enzyme molecule is unchanged and can be reused, but the products, so the small particles that have been produced from that large molecule, are released, and they're then able to be absorbed into the blood. So enzymes are really, really important in digestion. Okay. So we talked about how the small intestine is adapted for digestion, so it can do the best job it possibly can. But what can we do to give the best conditions possible for enzymes? Make them work as efficiently and as fast as we possibly can. Well, there are 2 factors that you need to know about. The first is temperature, and the second is pH. So we've got a graph here, the rate of enzyme activity on the y-axis there, and temperature on x-axis. And we can see that the rate of enzyme activity increases slowly as temperature increases. And then we get this quite fast increase here at a particular temperature until we hit the fastest possible rate of enzyme activity. And that is at our optimum temperature. So it tends to be, we run at about 37.5 degrees Celsius. So enzymes within the human body tend to work best at about that temperature. If you go beyond that temperature, the shape of the enzyme can change. So when you increase temperature, particles will move more, don't they? That's something, it's a kind of fundamental rule of physics. As you increase temperature, particles tend to move about more, they have more energy. And past the optimum temperature, those particles are moving so much that the shape of the enzyme becomes not as solid. And so it starts to break apart a bit and denature. And because the shape of an enzyme, that structure that forms that lock and key kind of mechanism, is so important, it means that it cannot work if it goes past the optimum temperature. Just seen a question saying, can the digestive enzymes also digest its own cell instead of food? The answer is no, purely because of that very specific shape of the enzyme. So the enzyme can only ever attach onto those large proteins and carbohydrates and lipids that come in as food. So it would never have the right shape in order to fit to its own cell. Okay, so we talked about how temperature affects enzymes, so now let's talk about pH. So pH is the acidity or the alkalinity of the environment. And the amino acids that make up enzymes, because remember enzymes are proteins, so they are made up of amino acids, they often carry a charge, so a slight plus or minus charge, positive or negative. That helps to form their structure because positive particles are slightly attracted to negative particles, and so it helps them to form that key shape. As you increase or decrease pH, that affects those charges. And so it means that if you're running at too high or too low a pH, the structure of the enzyme can be affected. So they're not going to be able to fit that lock and key mechanism. They're not going to be able to join to their substrate, their food molecule that we want them to. And so digestion won't be able to take place as fast. So you can see on the graph here, on the y-axis, we've got rate of enzyme activity. And on the x-axis, we've got pH. And this particular enzyme shown in this graph doesn't work very well at all at 2.5, so that's quite acidic. And then as the pH increases here, its rate becomes better and better. It works more and more efficiently and faster. And then at about 8, so that's quite near to neutral pH, that's its optimum pH, that's where it works best. And then as pH increases and becomes more alkaline, we see that rate decrease again until at 13 it's not really working at all. Would you call an enzyme denatured in an exam when the optimum pH is passed? So pH of more than 8. I don't think you could categorically say it was denatured until it's hardly working at all, so down in this area of the graph, but you could say that the pH is affecting the structure of the enzyme, so it's not working as well. So it's not working at its optimum rate, the best way that it possibly can. Okay, so another thing that we need to learn for GCSE biology is which enzymes break down which molecules. So we've talked about how enzymes work, we've talked a bit about the digestive system and where enzymes are working, but we haven't talked about the enzymes themselves. There's a few that you need to learn. Just seen another question about the last slide. Do different enzymes have different specific optimum temperatures and pHs? Yes, you are correct. So quite often in an exam, you'll get questions where they talk about an enzyme having an optimum pH or an optimum temperature, and then they'll ask you questions about what would happen to that pH to that enzyme at other pHs or temperatures. It's one of their favourite questions, actually. A really good example is, so at some hot springs in America where you get the really good, the really high temperature thermal springs, the natural hot springs, there are enzymes in there, so organisms that survive in those really hot temperatures. And their enzymes obviously work at a very high optimum temperature, and they're adapted to do that. Okay. Let's talk about this slide. So different enzymes break down different molecules. So we saw this diagram at the beginning of this lesson, but I've kind of filled in the middle a little bit more. So at the top we have a protein molecule. It's made up of lots of different amino acids. That protein molecules are broken down by enzymes called protease enzymes. Enzymes quite often have the suffix ase at the end of them. So it's a good way of remembering some of the names. So protease breaks down protein molecules into amino acids. An example of a protease enzyme is pepsin. Here's the starch molecule. Starch is a carbohydrate molecule made up of lots of different glucose molecules. In order to break that starch molecule down into individual glucose molecules, we use a carbohydrase enzyme. And an example of a carbohydrase enzyme is amylase. Same again for fats. They're made up of fatty acid and glycerol molecules. And lipase breaks down fat molecules into its separate fatty acids and glycerols with the help of bile, which again, we are going to talk about later. This is a fairly horrible looking table. which lists all of the enzymes that are needed, particularly for AQA specification of GCSEs, but a lot of these are included in other specifications as well. So we've got 3 carbohydrase enzymes here at the top. Salivary amylase, pancreatic amylase, and just amylase. So those are fairly easy to remember. They're amylase and they're named after where they're produced. So salivary amylase is produced in the salivary glands and works in the mouth. Breaks down starch to a 2-sugar compound called maltose. So it starts to break down the starch, but it doesn't quite get as far as glucose. Then pancreatic amylase works in the duodenum, part of the small intestine. That's produced in the pancreas, and it breaks down starch to maltose again, so it continues. Remember when we looked at the digestive system diagram, the digestion of carbohydrates starts in the mouth with salivary amylase, and it continues in the small intestine, and then finishes in the ileum. So amylase that works in the ileum finally manages to break down that maltose into glucose. So you've gone from starch, which is a really long carbohydrate molecule, to maltose, which is a slightly shorter carbohydrate molecule, and then finally to glucose, which is our end product. Okay, then there are 3 protease enzymes here as well. We talked about pepsin in the last slide. Pepsin works in the stomach. It's produced by glands in the stomach. It breaks down proteins and begins to break them down to amino acids. And we were talking about optimum pHs for enzymes. Well, the stomach is quite an acidic environment. So enzymes that work in the stomach have to have quite a low optimum pH. Then we have trypsin. This works in the duodenum, so the upper part of the small intestine that we talked about. Trypsin is produced in the pancreas. So when we looked at the digestive system diagram, we talked about the pancreas producing some of the enzymes that were needed for digestion. This is where that comes in. Trypsin also works on proteins and continues their breakdown to amino acids. And then finally, we move to the ileum, which is the lower part of the small intestine. And there we have peptidase enzymes that are produced in the wall of the ileum. They complete that breakdown to amino acids, which can then be absorbed into the blood. And finally, we have lipase enzymes that break down our lipids. Now, I don't know whether you remember at the beginning when we looked at the diagram of the digestive system, we talked about lipase or the digestion of lipids not happening until a bit later on. So the digestion of lipids happens in the duodenum, so it starts quite a bit later than our protein and carbohydrate digestion, which started in the mouth or in the stomach or the mouth or the stomach. Lipase is produced in the pancreas and breaks down lipids to their fatty acid and glycerol components. You might not have time to write all of those down now if you're making notes, but this will be available to watch later, or I can switch back to it at the end if needed. Okay, I'll give you a second just to keep looking at that one. I don't know whether you can see most of it on the slide. And then carry on. I'll have a quick look at questions. Are there enzymes with other functions other than digesting? Yes, there are. So enzymes are our catalysts of all reactions that happen in our body. So there are enzymes that work in all sorts of different roles. So producing, there are enzymes that work in respiration, there are enzymes that work in conversion of, say, glycerol to glucose to glycerol, glycogen to glucose to help us with our respiration, and lots of other reactions as well. Yes, carbohydrates, enzymes all have a unique shape. So all of these enzymes that we've listed now have their own unique shape that bind to carbohydrates such as starch or proteins or lipase. So each enzyme has a specific shape that is complementary to the molecule that it's designed for. Okay, main difference between the duodenum and the ileum is that one is higher up, so they're kind of two different parts of the small intestine. Okay, we've talked about bile a couple of times, but until now it hasn't really fitted to talk about it properly. You needed to know about some other things first. So bile is produced in the liver, stored in the gallbladder, and then released into the small intestine. And bile has two particular functions. So the first thing it does is emulsification. It breaks down large drops of fat or oil into small droplets so that lipase enzymes can work to break them down into fatty acids and glycerol. So if you've ever spilled oil into water, you know that it forms those kind of weird, kind of slightly shiny looking droplets. And those are very, well, they don't dissolve in water. They're very resistant to having anything done to them. And so lipase enzyme has quite a tough job of it. Those are hard to break down. And bile is our body's way of helping lipase do that. So it helps to break down the large drops of fat or oil into smaller droplets. And each of those smaller droplets can then be surrounded by lipase enzymes that can get a really good hold and really break it down into that fatty acid and glycerol. So that's one function of bile. The second function of bile is that it helps to neutralize stomach acid pH. So bile works in the small intestine, which is below our stomach. So the food has already passed through our stomach and then heads on down, and the stomach's a very acidic environment. And not all enzymes are adapted to that acidic environment. They don't like being in acid conditions, so they don't work as well. Bile works in the small intestine to help neutralize that stomach acid. It contains something called sodium hydrogen carbonate. Neutralizes that stomach acid, brings up the pH, and so provides an optimum pH for pancreatic enzymes to work in the duodenum. So if we go back to the last slide, which are the enzymes that work in the small intestine. So pancreatic amylase, produced in the pancreas. That works in the duodenum, the small intestine. Amylase works in the ileum. Trypsin as well, those protease enzymes, peptidase and lipase. So they all need that non-acidic environment, and bile helps to produce that in the small intestine, or in the duodenum specifically. So that's how bile fits into the equation. Okay, so next I've got a past paper question, but I think what we might do is just have a quick look back at this diagram of the digestive system and have a look at it again, knowing what we know now about enzymes and how they're working. So we start at the mouth, which begins the digestion of carbohydrates. And if you remember, we talked about salivary amylase that's produced in our salivary glands and starts breaking down carbohydrates from large starch molecules down to slightly smaller ones in the mouth. Our food then passes down the esophagus and into the stomach. And it's in the stomach that a couple of enzymes begin the digestion of protein. So let's check which enzymes it is that work in the stomach. It's pepsin that works in the stomach. So that begins the digestion of protein and starts some of the absorption, but it's quite an acidic environment, remember, so it needs enzymes that are adapted to that quite acidic, that quite low pH. From there, the food passes to the small intestine, the upper part of the small intestine, which is called the duodenum. So on that diagram, let's put the pointer on. You can see the duodenum's kind of here. And then the ileum starts all the way over here. So our small intestine is really, really, really long. And it's all curled up underneath our stomachs. All of this brown area here is our small intestine. In the duodenum, which is the upper part of the small intestine, the digestion of carbohydrates and proteins is continued, and we begin digesting lipids as well. So if we go to our table of enzymes, you can see that pancreatic amylase works on carbohydrates in the duodenum. Trypsin works on proteins, and then lipase works on our lipids with the help of bile, which we talked about before. Then we move to the ileum, and by now we've done quite a lot of the digesting. So now we're getting more around to the absorbing. So here we complete the digestion of carbohydrates, proteins, and lipids. There were a couple of extra enzymes here. So the ileum, amylase works in the ileum and does that final step in carbohydrate digestion down to those single glucose molecules that can then be absorbed into the blood. And peptidase completes the breakdown of proteins to amino acids. And lipids will finally be in their fatty acid and glycerol state. Okay, so then in the remainder of the small intestine and the large intestine, we have all of the absorption of water and the rest of all of those small particles that digestion has broken down the large particles into. Those are absorbed through those villi that we talked about, the small intestine, straight into the blood, and the blood can then transport all of those particles and molecules to where they're needed in the body. Okay, let's do a past paper question. So, lipase is an enzyme that digests fat. We've just talked a lot about lipase. Complete the equation to show the digestion of fat. Use the correct answer from the box. So fat, with the help of lipase, becomes fatty acids and glycerol. This is a 1-mark question, so we just need the 1 mark, the 1 word there. Name one organ that makes lipase. Again, 1 mark. The answer to that is the pancreas. So if we go back to our table, see that lipase is produced in the pancreas. Okay. Some students investigated the effect of bile on the digestion of fat by lipase. The students, 1, mixed milk and bile in a beaker. 2, put the pH sensor of a pH meter into the beaker. 3, added lipase solution. 4, recorded the pH at 2-minute intervals. 5, repeated steps 1 to 4, but used water instead of bile. Okay, so this is an experiment. When we're talking about questions like this, it's really handy to underline the keywords. So we've got investigated here, that says that's an experiment. The effect of bile, we've just talked about bile, on the digestion of fat by lipase. We know that lipase is an enzyme digest fat. As an enzyme, it's quite affected by temperature and pH. Sure enough, students have mixed milk and bile in a beaker and put a pH sensor of a pH meter into the beaker. They've then added the enzyme and recorded the change in pH. Suggest two variables that the students should have controlled in this investigation. 2-mark question, we're suggesting two variables. You could have had any of type of milk, volume of lipase, or temperature. All of those things are needed to keep that experiment fair and make sure that the only thing that you're changing is whether it's milk or bile, or whether it's water or milk and bile. This is why it's very important to read the question properly. And that they're measuring the pH. Okay, then we have a nice graph. This is the last part of that question. Ooh. Here we are. So let's have a look at this graph. When we're approaching a graph in an exam, it's important not to worry. That's the first thing. And also to make sure that you take notice of the axes and the units and also anything that the graph is showing before you even look at the question. So what have we got on the y-axis here? We've got pH. And the units are, pH is running from 7.4 at the bottom here, so that's near neutral. This axis doesn't start at 0. It's really important to notice that. And then it moves up to 9.2. So it's becoming a little bit more alkali at the top there. And on the x-axis, we have time in minutes. Runs from 0 to 16 minutes. Done about a quarter of an hour's worth of timing here. Our graph has two lines. We've got a solid line, indicates the pH change over time with bile. And then we've got a dotted line that indicates the pH change over time with water. Okay. Then we've got three questions. The first is, why did the pH decrease in both investigations? This is sort of quite an intuitive thing to have to work out. pH decrease means that the conditions are becoming more acidic. When lipase works on the fats that are in milk, it breaks them down into fatty acids and glycerol. Fatty acids themselves are quite acidic, as it suggests in the name, and so they're going to lower the pH as they're produced. So it'll become more and more acidic. Question 2, bile helps to digest fat. We just learned about this. What evidence is there in the graph to support this conclusion? Okay. So if we look at our graph, the solid line is with bile. And we can see that the solid line goes down more quickly than the dotted line. So the solution is becoming more acidic more quickly, which means that more fatty acids are being produced more quickly with bile. And so bile is digesting those lipids or helping lipase to digest those lipids more quickly. So the pH decreases faster with bile, so digestion is happening faster. And finally, suggest one reason why the contents of both beakers had the same pH at the end of the investigation. And it's true, if you look at the graph, you can see that although with bile the line goes down faster, they both hit that lowest pH of 7.6, which is about neutral. But with bile, that's hit at about 9 minutes, whereas with water, it takes the full 16 minutes to get there. Okay. Both beakers had the same pH at the end of the investigation because all of the fat within the milk had been digested by lipase, so the same amount of fatty acids had been produced. So they've both gone down in pH an equal amount. So that's how we would approach that question. Okay, let's have a look at some of your questions now, or some more of your questions, and see what we can answer. Frederica, did you spot any more that I didn't see?
I just had a few on the Q&A saying if you could please go back to the slide with different enzymes, that would be helpful.
Oh, okay, no problem.
And then I actually had a few little ones. So the first one is, does bile act as an enzyme or a catalyst?
Bile isn't really either. Bile is kind of a helper. So it's not an enzyme. Bile is kind of a liquid. And so I think the best way to describe it is a helper. So it helps by emulsifying those fats into smaller droplets, and then the enzymes can work on it better.
Cool. And then another question that came through was, do we need to be specific by which part of the intestine a process is happening within during an exam?
The answer to that is that it depends a bit on the question. I'm afraid you do need to know in general which parts of the body each stage of digestion takes place in. So particularly for carbohydrates, where digestion starts in the mouth, continues in the stomach, and then finishes in the small intestine, you do need to have kind of a knowledge of that gradual change. If you've got some of the longer questions where there's more marks available, there will be more marks for suggesting which areas of the digestive system are used at what point, and you're breaking down different molecules. So it is worth knowing, unfortunately. It might be worth making a slightly nicer revision aid than my table. My table contains all of the information there that you need to know, but that you could rearrange that and make it look a little bit nicer.
And then the last one for me on here at the moment is, do different enzymes have different specific optimum temperatures and pH?
Yes, they do. So enzymes that are working in the stomach, obviously the stomach is quite an acidic environment, so they have an optimum pH that's very much lower than most enzymes, and that's why bile is so important when you get to the small intestine, because bile also acts to bring up that acidic pH and create that more optimum pH in the small intestine that pancreatic enzymes needed, or enzymes that are produced in the pancreas need, because they can't cope in those really acidic conditions in the stomach, and they start to denature. So yes, different enzymes all have different optimum temperatures and pHs, which is why you get these kind of different sections in the body where they all work, and they're all really well adapted to where they work best. our body is always working towards keeping everything optimum, making sure that every process can work as fast and as efficiently as it possibly can.
Great. And there's another question that has come through asking, why is, in question 1, why is the glycerol not glycogen or glucose?
Question 1. Oh, yes. Okay. So there are lots of words that begin with G in biology. And they are all often spelt quite similar as well. So you have to be really careful with making sure that you know which one is which. So here, fats are made up of fatty acids and glycerol. Okay. Glucose is something different again. So glucose is the smallest part of a carbohydrate. So if we look here, our carbohydrates are broken down into molecules of glucose. Glucose is what is used for respiration in the body. So if you remember, respiration is glucose plus oxygen gives carbon dioxide and water, the opposite of photosynthesis. And so that's what glucose is used for. And then the final one was glycogen. Glycogen is something different again. So glycogen is how glucose is stored in the body when we've got more than we need for respiration. So sometimes when our cells are respiring, they're using up that glucose, but we've got a bit extra. And so our cells store it as glycogen. And then when we need to respire really fast, so say when we're going from a jog to a sprint, then they start to break down that glycogen and use that glucose. So those are 3 different words that begin with G with 3 very different definitions. So in this case, the right answer was glycerol, which is what helps to make up a fatty acid. But yeah, there's not really any way to easily remember them. They need to be memorized. And I mean, it might be quite handy to sort of make a mind map of words beginning with G in biology. Make sure that you know the difference. Okay, any other questions?
I don't think anything for the moment, but while I just have another look, would you mind carrying on with another question if we have one?
Yeah, that's fine. I'll just pull one in. So we've got another kind of more scientific one here that we can talk about. So it's quite a long one, but I'll pull it in now. Okay. Here's the first part. Have a read of that while I pull in the second part here. I'll put the diagram over here. Have a look at them both. And then finally, we've got a table as well. I'll put that underneath. Okay. First question. So I'll pop over on this side as well, so that we can kind of move everything around as we need to. Okay, so let's start from the beginning. Catalase is an enzyme. I'm going to underline the keywords here. Catalase, we know it's an enzyme, it ends in Ase, so we could have a decent guess at that. Catalase controls the following reaction. Hydrogen peroxide is converted to water and oxygen. Okay. Student did an investigation on catalase activity, and the method used was to put 1 centimetre cubed of hydrogen peroxide in a test tube, add 1 centimetre cubed of catalase solution, bubbles of oxygen are produced, and bubbles cause foam to rise up the tube. Then they measured the maximum height of the foam. So we've got the diagram here. It's carried out at 20 degrees C. We've got the height of the foam here. Then we've got the table of results, which for some reason extends 1 row below the end of the page. So we can see that the temperature increases in 10 degree increments from 10 to 20 to 30 to 40 to 50 and 60. And then we've got the height of the foam in test 1, test 2, and test 3, and then the mean at different temperatures. Why did the student carry out the experiment 3 times at each temperature? Tick 1 box. So is that to make the experiment more fair? More accurate, to prove the experiment was correct, or to show the experiment was more repeatable? Okay. So whoever thought that to show the experiment was more repeatable was the answer, you're correct. Okay. Let's pull up second and third part of the question. So a lot of these are about experimental technique. The student thought one result was an anomaly. Circle the anomaly in the table above. Do we know what an anomaly is? So an anomaly is a result that looks very different from the others. It's an outlier. It doesn't fall within the kind of average result. So if we go back to our table, we want to circulate the one that we think is an anomaly. Okay, so have a look at that table. Got lots of different results in here. The only one that stands out as being very different is this 0. It's very unlikely in an experiment that you would have no reaction at all. And so the fact that you've got a 0 there says probably that that particular part of the test didn't work, rather than that that's a decent result. What did the student do with the anomalous result? So what should we do with anomalous results? Anomalous results should always be ignored. Let's put did not use it. So you shouldn't use it in any analysis that you're doing after the experiment. Okay, we've got another tick box here about the conclusion. what conclusion can be made as temperature increases? So let's have a look at the options before we look back at the table. So we've got decreases the rate of reaction up to 30, decreases the rate of reaction up to 40, increases the rate of reaction up to 30, increases the rate of reaction up to 40. So let's move back to our table. We talked about up to 30 and 40 here. So if we have a look at our mean column, the maximum height of foam has increased up to 30 and then starts to decrease a little bit. That would be the general trend. So if we go back to here, we can say that it increases the rate of reaction up to 30 degrees C. It decreased again before it went to 40. Okay. Then there's a part E. At which temperature was catalase denatured? So we know that catalase is an enzyme, and so here we're talking about at a certain temperature, catalase couldn't hold its structure anymore, could no longer bind to its substrate in that lock and key mechanism that we talked to, and denatured. Okay, so if we go to our table, and we need the last row of the table for this question. So here, see we have a whole row of zeros at the bottom here. That's where the enzyme just wasn't working anymore. And so that's what we need to tick. So that's where it had completely denatured. Okay, so that's that question. Were there any more questions?
Just having a look now. I don't think so. I think we've only really got about a minute and a half left anyway. So it's probably a good time for me just to say that if you really enjoyed the session today, which I hope you did, then we'll have some more biology sessions this week. So look out for the timetable on the My Tutor online school to know exactly when those will be, and also what topics we'll be covering for those sessions. And just to say, like we said at the beginning, that this session has been recorded today, so you will be able to find a link to that. And the best way to make sure that you're updated with not only what sessions we have to come, but also what videos you can watch on the My Tutor online school is to be signed up to the online school. So make sure that you've done that. And I hope to see lots of you guys tomorrow for another full day of tuition. So that's everything from us. Thanks, Charlotte. Thanks, guys. See you soon. Bye.
Bye.